Now we're going to get to some definitions. The first definition is probably the trickiest one, may even be the trickiest definition we have in this course. And that's, what is the definition of a gene? Well, it says here on this slide, and I'll take the rare step of actually reading it to you. It says here that a gene is a region of the DNA that's made into protein. And that's true, but it turns out that molecular biologists use the term gene in a broader fashion. Now, it's true that it includes those segments of DNA that are used to make proteins. So a segment of DNA or a region of DNA that's made into a protein is definitely a gene. But there's also regions of DNA that we'll see later on in this course, a little bit, but mostly in other courses, that are regulatory in nature. So they're little segments of DNA that may or may not be made into RNA that come around and regulate various parts of this process. Well, those segments of DNA that perform the regulatory function are also typically called genes as well. So a better definition of a gene, but one that we don't need to learn for this course, a better definition of a gene is a region of DNA that does something. Well, now you see why it is that we didn't use that definition. That's not much to learn, okay? It doesn't tell you very much. Almost always, what that region of DNA does is get made into a protein. But there will be other cases where that DNA has some other function, and we're still going to call it a gene. But for the purposes of this course and the purposes of 90% of the world of molecular biology, a gene is a region of DNA that's made into a protein. The RNA molecule that's responsible for being copied and carrying that information outside of the nucleus to the cytoplasm where the ribosomes can act on it is called messenger RNA. And you don't have to think too hard to figure out why it is that we call this messenger RNA, because it's the RNA that's carrying the code for a primary sequence of amino acids in a protein. So in the process of translation, that RNA transcript is translated into a protein primary sequence, the sequence of amino acids in the protein. Shown here is AA1, the first amino acid, AA2, the second amino acid, AA3, the third amino acid, and so forth. And remember that when we string amino acids together in an ordered sequence, we call that the primary sequence of the polypeptide or the protein. Going back to transcription then, DNA makes RNA. And this is shown in a way that we can actually see with our own eyes in this photomicrograph. This is a photomicrograph of DNA taken from a living cell and being asked to make an RNA transcript. So we're actually seeing the process of transcription. One thing you need to know is that when a gene is very busy, it's being transcribed at multiple sites. So there's a lot of different places where this transcription begins and continues. So which way is the process going? Is it going from left to right, or is it going from right to left? We'll hold on to that question, answer it in a future slide. Transcription makes RNA. So that's part of our central dogma. DNA is made into RNA. The process by which DNA makes RNA is called transcription. So the first draft of RNA is a direct copy of the DNA. The first draft of RNA is a direct and perfect copy of the DNA. But like anything, like this video, before I started hacking away at it, it has to be edited down to the final version. So I leave a few mistakes in just so you can see I'm human, but as I edit down these videos, I'm taking maybe 2 hours of video and editing them down to maybe an hour and a half, depending on how many mistakes I make, of final video content. And so that editing process is also happening to the DNA transcript that we're calling messenger RNA, also happens to the messenger RNA before it can be made into protein. The enzyme which is responsible for turning DNA into RNA, Not surprisingly, we know it's an enzyme because it ends in ASE. We know what it does because it's called RNA polymerase. So RNA polymerase is an enzyme which makes an RNA polymer. And we're taking RNA monomers and stringing them together into an RNA polymer by using that ribose phosphate backbone. The enzyme that does that takes the RNA monomers and strings them together into a messenger RNA polymer, is called RNA polymerase. The site at which we begin doing that is called a promoter. That's the site where RNA synthesis is started. And then RNA synthesis has to end somewhere. We can't possibly afford to transcribe all of the DNA in the cell at all moments in time. That's just not possible. As we begin to look at the size of DNA, I talked about a little bit in Module 3, but as we look at the size of DNA, that's just not going to be possible. So we only want to work on those segments of DNA that are important to the cell at that moment in time. I call those switches that are turned on. They're molecular switches. So we're transcribing only one gene or only a suite of a few dozen or a few hundred or a few thousand genes in a particular cell at a particular time. And each one of those genes has a promoter region, which is where the RNA polymerase lays down and starts making the RNA, and then a terminator region, which is where the RNA polymerase lets go and quits making the messenger RNA. So now we have the tools to answer the question that we asked earlier. Part of the reason that it was a hard question is because we didn't have all the tools for understanding the whole process, but now we can see that the correct answer to the question is A. Transcription is moving from left to right. I know this because as I move from left to right along this DNA strand, the mRNAs get longer and longer. And that means they spent more and more time with the RNA polymerase. So the earliest ones are the shortest mRNA strands, shown at left, and the oldest ones are the longest mRNA strands, shown at right.